All right, so we're going to kind of continue what we were talking about yesterday. A little bit more to it than we talked about yesterday. But this idea of crossing different organisms, right? Kind of a review here. We use the idea of a Punnett square, right? We had the square, and the square let us statistically analyze what was being possibly made. So, for example, if we said a female, right, had, say, I don't know, she had type AB blood, right, should form an egg with an A allele in it and a B allele in the other egg, right? Statistically speaking. So one half of her eggs would have an A allele, and one half of her eggs would have a B allele. We said that males, for example, if we're talking about AB blood, we have a male here. Let's say he is type A blood, but he has an A allele and an O allele. And then statistically we said that what? That he would have an A allele in one half of his sperm and an O allele in the other half of his sperm, right? Then we combined these and said, statistically, what would we see? And statistically, what? 1/4 of the children would have type A blood. One half of the children have type A blood, right? This is an A blood type, this is an A blood type. 1/4 would have type AB, and 1/4 would have type B, right? Statistically, does this mean that it will be that way if they have 4 kids? Does this mean that 2 will have type A blood? No. Statistically, though, that was what we're saying. That's the most probable outcome. All right. Now we're going to get in this idea. This is a one feature difference. Okay. Here we're talking about one gene for blood type, but we're going to expand our definition slightly because we want to talk about what if we have 2 different characteristics, right? 2 different things. Let's say, and I'm going to show you an easy way of determining this. Do you remember we talked about last time where we just take the letters and move them around real easily? Okay. Let's just throw some things out here. Let's say there's 2 features. Let's say that white, let's see, another one. Green, big G is green, that's going to be dominant. And a little y will be recessive. So let's just say one feature is leaf color. Okay, this is leaf color. If someone has a G allele that's dominant, a Y allele is recessive. Let's also say there's a feature of height. Okay. If they are tall, that will be a big T, and that's going to be a dominant. If they are short, that'll be a small s. Okay? So we have 2 different characteristics we're looking at on this. Now, one thing with this is these we're assuming are on different chromosomes. Okay, we're assuming these are going to be on different chromosomes. We'll talk about linked genes next time, but right now we're assuming these are on different chromosomes. So let's just say we have a female and she has 2 of the G alleles for color, and she has a tall and a short allele for height. So she would be what? Color-wise should be green. Height-wise should be what? Tall. Now, what kind of gametes could she make? What kind of possible egg combinations could she make?
Green and tall and green and short.
Green and short. What ratio?
3 and 1.
Not 3 and 1. Now, there's an easy way. I'm going to show you the easy way of doing this, and then we'll explain why that is in a second. But let's just use, remember FOIL from math? With FOIL, if this was numbers, you would do it this way, right? Use a color, different color. You do this way, and you then do this way, right? You do this way, and that way, right? Okay, just FOIL it. Okay, we'll explain what that means here in a second. But if you use FOIL, you'd have one that was green and tall, one that was green and short, one that was green and tall, and one that was green and short. 2 and 2, exactly. Okay, so you take them and you do FOIL with them, and that will give you the possible outcomes. Now that's a nice, handy way of doing it, but it kind of ignores the point here, what's going on a molecular level. So let's reset this here. Remember we talked about how when we have the chromosomes around the gene, or excuse me, we have 2 homologous chromosomes in meiosis, right? And those can go on either side. It's randomized. Okay, this is kind of describing that randomization. How? Let's say this female was green and yellow and had a tall and a short allele. All right? Now, let's just say this was her nucleus at the time here. Two sets of homologous chromosomes. On this one, she would have a G for green and a yellow for yellow. On this one, she would have a tall and a short, right? If these were to split, right, what kind of gametes would they form? One gamete would have green and tall, right? The other one would have what? Yellow and short. Now, remember we said that which side these wind up on is randomized? Okay, well, let's just say these two flipped. Now let's say instead of being tall here and short here, just how these chromosomes wind up around metaphase, it flips. Now, when these separate into two, what would it form? Green and short, and yellow and tall. All right? Now, let's go back and do that FOIL with this one up here and see if we can see that. Green and tall. Do we have green and tall here? Yep. Green and short. Yep. Yellow and tall. Yep. Yellow and short. Yep. So if you want to determine the possible gametes that can form, do FOIL. But the story behind it is during metaphase, you know, how these chromosomes line up on that metaphasic plate, that metaphasic line, that can vary. And this is just a nice mathematical way of looking at it. All right? Okay. So, let's say we have a male and a female, and the male is green, has a green allele and a yellow allele, and he also has a tall allele and a short allele. The female of this plant, we'll call it a plant, similarly has a green allele and a yellow allele, and a tall allele and a short allele. Okay, this is what we call a dihybrid cross. Di meaning two. Remember we said if we have two different alleles, it's a hybrid, right? So there are two different alleles for two different genes, hence the di. The hybrid comes in because we have two different alleles. Okay, so it's a dihybrid cross. All right, so let me draw my Punnett square out here really fast. All right. Now, the male. What kind of gametes could that male make? Green and tall. What else? Green and short. What else? Yellow and tall. Yellow and short. And again, you just did that FOIL thing, right? All right. Now, how about the female? What can she make? The same thing, right? Green and tall. Green and short. yellow and tall, and yellow and short. And again, all we're doing is foil. But that foil represents what's happening during metaphase, where those chromosomes can go on either side and segregate to any other side. So the point is we're really looking at what's happening with the cell, but to keep track of all that stuff, just use foil. All right? So now, and again, before I go into this, this is a statistical argument. Okay? Does it mean that you're going to get these exact ratios every time? No. Okay? It's a statistical argument that what we expect to be the most likely case. Does it mean, for example, you flip a coin 10 times. Statistically, what do you expect it will be heads? 5. And 5 will be tails. But I guarantee you flip a coin, sometimes you're going to get 4 heads and 6 tails, right? You know, just because you expect a statistical outcome does not mean you will get that exact statistical outcome. All right, so these represent what? What do each of these circles represent? Gametes, correct. Possible sperm combination, possible egg combination. All right, so we're just going through the cells here. Green, 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 tall, tall. Green, green, tall, short. The combination having 2 green alleles and 2 yellow alleles, or excuse me, 2 green alleles or 2 tall alleles, we called that the what of this. This was an individual, what would we call that? The G. Genotype, correct. Now what color would this person be? Green and tall. That's his what? Phenotype, correct. This would be green and tall, right? That's the phenotype. What's the genotype? 2 G alleles, 2 green alleles, a tall allele, and a short allele. That's the genotype, all right? So now we have green, yellow, tall, tall, green, yellow, tall, short. So let's see what we got here. Of these 4, what percent will be green? No, green, green. It's heterozygous, still green, green, so they'll all be green, right? So all will be green. How many will be tall? How many will be short? Tall, tall, 2 tall alleles, that's dominant, tall. No, they're going to be tall. So it says tall, 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 and tall. So they're all going to be tall. All right, let's go to the next one. Green, 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 tall, short, green, green, short, short, green, yellow, tall, short, green, yellow, short, short. One thing I'm doing here, why am I putting one letter first and the other one? If you have 2 different alleles, you always put the dominant allele first. So here we had green and short. Here we had green and tall. Tall would take precedence over short because we said the tall was dominant, right? Okay, that takes precedence. All right, so green and tall, green and what? Short. Green and tall, and green and short. So 1/2 are going to be green. Well, we won't put it here, we can't fit it all in here, but point is 1/2 will be green and tall, and 1/2 will be, excuse me about that. 1/2 will be green and tall, 1/4 will be green and short, green, tall, green, short, green, tall, green, short. Okay, that's right. 1/2 will be green and tall, and 1/2 will be green and short. You see that? All right. Green and tall, green and short. Green and tall, green and short. All right. Now I'm going to fill the bottom 2 in because I think you've got the idea. I don't want to, stretch this out too much, but have you guys been getting the idea? And I'm sure you've done a lot of this in high school, or some of you have.
Is our next test going to be our midterm?
It would probably be counted for midterms if we have it by then. I'm not sure. We haven't gone in the semester long enough for me to know what the next test will be. Yeah, because I kind of like seeing how fast the class is going on it, and then I can tell you when we're having a test. Turn the light on here. All right. Because yeah, that's the problem. Is if we're not if the entirety of the class is not keeping up the concept, then I might need to delay it a little bit longer. So I'll know a little bit more by next week. And the next week I should be able to have an idea. All right. So when we do this, how many of these are going to be green and tall? Green and tall are our two dominant characteristics. 1, 2, 3, 4. All these are green and tall, right? So we have 4 that are green and tall. Let me erase this right here. So what are green and tall? 1, 2, 3, 4. This row, 4 is there. Just so we don't get confused, I'm going to come in and just cover them up so we don't recount them. That's going to save us a little bit of time. All right. On this row, how many are green and tall? Green and tall? Is this going to be green and tall? No. Is this? Yes. And is this? No. So 2 will be green and tall. Let me get rid of those while I'm here. Okay. Will this be green and tall? Yes. Will this be green and tall? Yes. Will this be green and tall? No. Will this be? No. So 2 of these will be green and tall. I'm just going to get rid of those. All right. Will this be green and tall? G and Y, T and S. Yes. Let's get rid of that. G and Y, S and S. No. And will this be green and tall? I'm sorry. Okay. Green and short, so that's not 1. Y and Y, so that's going to be yellow and tall, that's not it. And yellow and short, and that's not it. So 4, 5, 6, 7, 8, 9. So we got how many? 1, 2, 3, 4, 5, 6, 7, 8, 9, right? We agree with that? So 5, 6, 7, 8, 9. 9 of these are green and tall, right? All right. How many are going to be green and short? Well, green and short, right? Green and short. Green and short. Yellow and tall, that doesn't work. Yellow and tall, that doesn't work. Green and short, that works. Yellow and tall, nope. Yellow and short, no. All right. So what's the next one here? Yellow and tall will be what? 3. And then short and short will be 1. All right, so we've seen all this. Will be 1. All right. Now, this is a classical example you've done before. I might not give you a dihybrid. I might give you something entirely different. I might give you one that's heterozygous for one and homozygous recessive for the other. So just because I've done, you've seen this ratio before, I can give you an entirely different one. Are you comfortable enough with it if I give you a different one? All right. So we have 9 in all this. So let's say you did this, and you got 1,000 plants from this. How many of the 1,000 plants would be green and tall? No. 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 And that's the problem. When you hear the ratios like that, no. If you have 1,000 plants, how many of those 1,000 plants will be green and tall? How many will be green and short? How many will be yellow and tall? And how many will be short and short? Or excuse me, yellow and short. So we said that this is a ratio of 9 to 3 to 3 to 1, right? All right? How you do it? You add up every number in your ratio. That's your denominator. Your numerator is the specific number you're looking for. 9 plus 3 plus 3 plus 1 is what? 16. How many is going to be green and tall? 9 over 16. Calculator out here. 9 divided by 16 is, percentage-wise, 56.2%. Okay, what's 56.2% of 1,000? 562, correct. So I would expect 562 green and talls. Okay? What is 3 divided by 16? Three divided by 16 is 18.7%. So I'd expect how many to be green and short? 187. Correct. And then the other one is what? 1 divided by 16 is 6.25, or about 62 would be yellow and short. Okay, so I'd expect 562 to be this, 187 to be this, 187 to be this, and 62 to be that. Conversely, I could also give you a number and ask you to determine a ratio. Okay? So for example, you have got 1,000 plants. 750 of these plants, this is a monohybrid cross. Okay, let's not worry about this dihybrid cross. Let me erase that so there's no point of confusion. Let's say you have 1,000 plants. And 750 of these plants are yellow. 250 of these plants are white. Based on that, what phenotypic ratio do you have? 3 to 1. Everyone see how we got that? Add these two up. 1,000 divided by 250. 750 divided by 1,000. One is 1 to 3. 3 to 1. You guys see we already did that? Okay. All right. Another example. You probably throw something at me when I give the example because everyone hates this example, but I'll still give it. A female who has type B blood but has a genotype of BO is married to a gentleman who has type A blood that has a genotype of AO. And let's just say that this follows a Brady-Bunchian type of genetics, in which case we got 8 kids here. I know it's more than the Brady Bunch, but let's just say we have 8 kids. The lady met this fellow and they had 8 kids. What percentage of the children, excuse me about that, what number of the children would you expect to have type O blood? That's the kind of question you could be asked. A male has, it doesn't really matter male or female here. The female has type B blood, and she has a genotype of B and O. A male has type A blood, and he has a genotype of A and O. And they have 8 kids. Statistically, how many of these 8 children do you expect to have type O blood? 2? That's correct. Didn't we see how we got this? Let me work it out. All right, good deal. So, Punnett square. We got one difference here, right? The O is recessive, the A and B are dominant. When A and B are combined, they're codominant. All right? So, the female will put up here, she has B and O. Hope she takes a bath soon. It's a joke. The O. Oh, tough crowd today. All right, the guy has an A and an O allele, right? So we do this, and we have an A and a B, an A and an O, a B and an O, and an O and an O. So what percentage of them will have the O alleles, the 2 O alleles, or type O blood? 1 out of every 4. Or could we maybe say 25%? All right, so 25%, or as he said, 1 out of 4 would have the 2 O alleles. What's 25% of 8? 2. Statistically, we would expect 2 children to have type O blood. But again, I go back to that flipping of a coin idea. It may be 1 out of 8 or 3 out of 8. Theoretically, it could be 8 out of 8. Because these are what we call independent events. Statistically, though, the most likely outcome we would expect from this data would be an outcome where 2 children have type O blood. How about I give one more example here? And this is another question everyone always asks about blood type. Question is, can a male and female that are type A and B blood, can they have a child that's type O blood? Yes. Someone asks me that usually every other semester. And yes, you can. All right.
They have type AB.
No. But actually, when I told the story a few minutes ago, in my when I took introductory biology, my biology professor told the whole class a story. They had like this semester before. Anyhow, someone asked, I believe this is how it went. I have AB blood, and my child has type O blood. How can that be? It can't. Except something called the Bombay phenotype. But aside from that, it can't be. Because you can't. If the male has type AB, if one parent has type AB blood, the child cannot have type O blood. It just doesn't. The genetics of it just don't work out. For example, let me just erase this. There is a caveat to this. There are things, for example, the Bombay phenotype, which can make blood look O in a coagulation test. So theoretically, it can, but that's something else entirely. Okay, let's just say the male is type AB. And let's just say the female is type O. That wasn't the case in this situation, but we'll do the numbers. AO, AO, BO, BO. 1/2 would have type A, 1/2 would have type B. So if a male was AB, No way to have a kid with type O blood. Because he's going to contribute either an A or B allele.
If both parents are AB blood, then the kid can no matter what be AB.
If both children, good question. Let's work it out. You said if both parents were AB blood, right? Let's see. Okay, let me ask you a question then. Let me do this. Let's use the same number of kids here, and let's say both parents are AB blood. What number of those kids will have AB blood? Well, let's work it out. Let's work it out. A and A and B, A and B, and B and B. 4, correct. 4. 4 of those kids would have type AB blood. All right, any questions on that? You want one more example, or do you think you're good? Another example, all right. And I'm not going to do this one. I'm going to give you the numbers on this one, and you guys will work this one out. We'll do a couple examples the other direction in a second. All right, let's say we have a we have 2 parents that are B and O. Okay? What number of the kids will have type B blood? What number of the children will have type O blood? Let's continue assuming we have 8 kids here. What number of those kids will have type O blood? What will have type B blood? Phenotypically, not genotypically, phenotypically. Okay, give me a few more seconds on that. All right, how many of the kids will have type B blood? 6. Everyone see how we got 6? That's correct. And then 2 will have type O blood. Again, for the people that are following this on YouTube, let's talk about this. We have B and B, B and O. B and O, and O and O. Type B, type B, type B, type O. So we can say that 75% will have type O blood, 25% will have type, 75% will have type B blood. 25% will have type O blood. 75% of 8 is 6. 25% of 8 is 2.
So, if both parents had O, all the kids have O.
Correct. Correct. If both parents have type O blood, and I put an asterisk next to that, let me say it this way. If both parents had 2 O alleles, then yes, it's not in the cards, so to speak. And the reason I'm being specific about that is there are situations where you can be genetically, say, like a type A blood, but phenotypically, when you measure it, it looks like type O blood. Okay, we talked about the Bombay phenotype. So, barring that rare example, yes. Okay. All right, that's all I really wanted to cover now. I think that'll pretty much wrap it up. I want to get the dihybrid thing. We'll finish the linkage groups and everything else next time. Are you good on these? Okay. For the test, what you have to be able to do is if I give you some numbers, you should be able to backtrack from numbers to develop a ratio. Okay. Well, let's just say, let's do an example here. Eraser. let's say you have 1,000 plants, right? And 500 of these plants are blue, and 500 of these are white. What ratio is that? 1 to 1, right? Okay. What kind of cross, what kind of genotypes of the parents could yield that combination? Well, let's look. Let's say we have one parent that is blue and white, and another parent that is white and white. I'm sorry? No. Let's work. We'll do both heterozygous in a second, and this is it. But we'll do what you're saying. We'll show those both. Oh, actually, let me do this one, then we'll do yours, and then we'll talk about it. All right. So if one parent was blue and white, what color would it be? Blue. If this was white and white, it'd be white. Okay. So let's do it right here. Blue and white, white and white. Blue, white, white, white. Blue, white, white, white. Blue or white? Blue, blue, white, white. Well, one parent, well, yes and no. One parent would be homozygous recessive, having 2 recessive alleles. One parent would be heterozygous, having a dominant and a recessive allele. Now, let's just run your numbers here. What if we had this? So let's just see the outcome if we had 2 heterozygotes here. Okay, B and B, blue. B and W, blue. B and W, W and W, white. So 1/4 of this would be white. 3/4 would be blue. So if these numbers, instead of 1,000, 500, 500, what if I said 1,000, 250, and 750? 750 being blue and 250 being white. Okay. So those are the kind of numbers you're going to have to run. I'm not going to give you like weird, like 5,268 of these and 3,298 of those. They'll be round numbers like 1,000, 2,000. I'm not here to trip you up. I'm here to make sure you understand the concept, not that you can plug a number in a calculator. So there'll be round numbers out of 100, out of 1,000, stuff like that. All right, are we good to go? Any more examples, or are we good with this concept? Tell you what, think about it over the weekend, and then I might start next class with a few more examples with everyone to see where we're at. All right? All right, then have a good weekend. See you Monday. Have a safe weekend.